Okay, so for the beginning, what I'd like to do is begin by justifying C-PAC and why we use it, or why we develop it, kind of the need for it. So I apologize that some of these things may be a little bit redundant with things that y'all have seen already if you were at the Brain Hack, but I think that'll be okay. Okay, fantastic. So just really quickly, I'd like to again start with explaining what I mean by connectome. So C-PAC is specifically the configurable pipeline for the analysis of connectomes. So we focus exclusively on connectomes currently. You'll notice that some things we're starting to call C-PACs with an X. And that's because in the future, we're going to start doing more than just connectomes. In the future, we're going to try to do more volumetric analyses, as well as do task-based analyses. We're adding this in. So we plan on expanding it a little bit more. But our primary focus in my research and the research of my colleagues is in, what is this? Let me try that real quick. Is with functional connectivity, is with functional connectivity derived from resting state fMRI. So we tend to focus mostly on functional connections in the brain. And again, I'd like to caveat that when we talk about functional connections, what we're talking about is correlations between physiological measurements that we're making in the brain. Right? So that doesn't imply that there's actually a wire connecting those regions together. Instead, it just means that those regions tend to work together, to collaborate together to perform tasks, right? And so we don't know if it's that there's some external generated or internally generated thought that is simultaneously affecting both of those regions, and that's why they're up, or if there's a causal relationship between them. We don't know any of those things. Mostly what we're measuring are correlations between them. But what we do know is that these correlations that we measure are highly sensitive and highly specific to disease. And also, we're able to use the resting state connectivity to understand a lot about inter-individual differences in cognitive abilities as well. So they have a lot of power in terms of our ability to study cognitive processes. Even though you wouldn't necessarily think so because somebody's lying in the scanner and not performing a task, right? But I'll explain a little bit of that to you. But so when we talk about the connectome, we can represent it in a lot of different ways. So this is, so here, this is kind of how is sort of how we would really like to be able to image or to construct a connectome, where we're really talking about these fiber connections between brain regions, and we're actually mapping things to the true anatomy. We can understand it in a way that a neuroanatomist would understand it. We're not quite there yet, but that's kind of our hope. So the with a connectome, it's essentially, we can construct it as a graph where the nodes of the graph are brain areas. So these are regions of the brain that are maybe functionally homogeneous or anatomically homogeneous, right? So if you're using a Brodmann atlas to define the brain areas, maybe they have similar cytoarchitecture, right? If you're using another atlas, maybe they have different fingerprints. But there's some reason why we believe that these areas form kind of a distinct unit of the brain. And we have, between those areas, we have edges that represent their connectivity. So for functional connectivity, that edge is indicative of a correlation between physiological processes. If it was structural connectivity, it would actually be, a biological BTI. Right? But that's sort of the basic idea of what a connectome is. The connectome can actually exist at multiple scales. We tend to study it at the macro scale when we use MRI because we're limited to the resolution and the physiological processes we're doing. But that's actually very okay, right? Because the processes that we're trying to study at the systems level probably map better to that scale than they do a cellular level. If we try to understand speech at a cellular level, that's going to be really complicated. So at least we could try to understand it at the macro level and then slowly move down. So that's where we really study. I wrote a review paper for Nature Methods on connectivity, and it was right around the time when the the Obama Brain Initiative started, And that was very much focused towards like cellular level connectivity. And their definition of connectome is, you know, cells and synapses. And there was a lot of criticism that come up. It's like, oh, well, it's not a connectome at this level, you know. But then I was trying to explain to them, well, you can't measure a connectome in vivo, in a living human being, and you can't study, you know, a lot of the processes that we're interested in something that you can map the connectome in, like a fish or whatever. So, but I guess the point is at the level that we're interested in, these areas are fairly big, that what we're studying. Maybe you can imagine them as a patch of cortex that's on the order of a square centimeter. So we'll skip that part and go to this one. So what are we trying to learn from these connectomes, right? So I mentioned we could study cognitive processes in connectomes, and how we would actually do that with resting state data is we might give people assessments outside of the scanner, right? So we may assess their working memory, we may assess their personality traits, you know, 5-factor neo, their positive and negative affect. We can measure all of these different things about them, their reading ability, things like that, and use that as a covariate to look at their connectomes and try to understand what variation in the connectomes seems to be correlated with things like people's verbal working memory or whatever abilities, right? And so we call that characterizing inter-individual variation in connectomes. So the idea is that individuals have some variation in their phenotype, and we can map that variation in their phenotype to variation in their connectome, right? A nice thing about that type of measure is that we can, you know, rather compared to a task measure, being able to do the task in the scanner at the time, is that we can measure a whole lot more outside of the scanner. So for example, with a Rockland sample, we have on the order of, you know, 20,000 variables that we measure on every individual. And it's, you know, so everywhere from, you know, administered questionnaires, physiological tests, cardiovascular fitness to interviewed structural interviews like SKIDs, WASI-Wyeths, things like that. So there's a very broad array of variables that you can correlate with the connectomes and try to understand people both in pathology and in healthy individuals. But probably where we hope that resting state may get the biggest effect or have the biggest impact, and where it actually comes from, is the study of psychiatric disorders, right? So the term resting state, when Bharat Biswal made his initial discovery about connectivity, functional connectivity in 1995, the term resting state was already widely used. So there's a lot of consternation about the name resting state fMRI, because obviously you're thinking about something, right? I mean, there's spontaneous thought, there's physiological processes, your heart's still beating, you're still breathing, you know, all of these things are going on. So it's not like the body is not doing anything, right? Even though originally when they were doing some of the scans for Bharat's work, he was actually looking at resting state as a baseline state where there shouldn't be, where anything you measured in the MRI signal would be noise, right? But he got it wrong. I mean, well, he didn't get it wrong, but that assumption was, you know, of course, a very kind of naive. But the basic idea and the basic beauty of resting state, particularly clinically, is you can put a person in the scanner and just scan them, right? You don't have to ask them to do anything. You don't need any particular level of compliance. You don't need to guarantee that they have a certain level of cognitive abilities, right? I mean, you can have somebody that's unconscious in a coma lying in the scanner. You can have somebody that, you know, is anesthetized lying in the scanner. So you don't have to worry about patient compliance or when you do it, and that's a very nice thing. Also, when you're doing tasks to study pathology, you have to make sure that your task actually matches the pathology, right? So you may get a null result just because, well, you chose the wrong task. And you know, there's a lot of good reason why we can do things like study, you know, motivation and ADHD, right? I mean, we can use the symptom space to hypothesize these tasks, but you could get it wrong and you could design it incorrectly. And so if we just use resting state and we have signal in resting state, we no longer have to worry about that. That no longer confounds us. So specifically, when we're studying tasks, we're interested in, what we really like to be able to do with resting state connectomes in pathological states is identify biomarkers. So with a biomarker, ideally we have an objective measure of whether or not an individual has a disorder or can index the disorder, right? We can tell how far along they are, ideally. And importantly, hopefully we'll be able to use resting state eventually to be able to make prognoses and to give people, help understand their outcomes, but also use it to apply medications to say, okay, well, based on your pattern, you're going to get better with venlafaxine rather than escitalopram. not going to say that word. But anyway, so that's another big thing that we're interested in. But also in the clinical bent, we've kind of talked about this before, but if y'all have been following the DSM-5 and currently staying current in matters of psychiatry, you'll know that there's a lot of consternation now about these categories that we apply to disorder in psychiatry, right? And so those categories are very useful for communicating to other people what a disorder is, and also for helping to find the right treatment. But the thing is they actually really haven't helped with biological psychiatry, right? So there's an article that pretty much says, why hasn't biological psychiatry or, in parentheses, neuroscience done anything for understanding diseases better? And the reason we think that this is that these categories have so much heterogeneity to them that they don't map to the actual biology very well. So, to give you two examples, 100 years ago, cancer was a non-specific disease. If you said somebody had cancer, it just meant they were sick, right? And now it's broken up into like hundreds of things, right? So we definitely have this tendency to take things and break them down as we understand them. And once we get to the physiological processes of cancer, now we can call it exactly like, B-cell lymphoma, or I'm not very good with cancer terms, so I don't know if that actually exists, but something of that nature, right? And so if you look at psychiatry and how we apply these things, there's syndromes, which means there's no underlying biological lesion that's been identified, but also they tend to be baskets of symptoms that we apply together. So for example, in order to be diagnosed with depression, you need 5 to 9 symptoms, and they have to have persisted for 2 weeks. That means there's 2 separate people that both have depression that only have 1 symptom. So there's a lot of heterogeneity there. So also we'd like to be able to use functional connectivity and biological measures to be able to split these groups, right? To be able to look even deeper and to be able to parse, say, depression or ADHD or ABIDE into multiple subpopulations so that we can maybe relearn what good classifications are. But I'm not going to go too much more into this, but I just want to kind of give you a little bit of an idea of the types of research that people are doing. And one consistent thing across all of these pursuits is they require a whole lot of data, right? So if we go back, one of the things that we're actually is good about now is that there's actually a whole lot of data that is available online that we can use, right? So these are very large data sets. Each one of these is at least 1,000 subjects, maybe even more. And these are very large data sets that are shared. Some of them are more phenotypic rich than others. But now we are starting to amass the data that will allow us to start answering some of these questions, where we have enough variability in both the phenotypic measure as well as enough individuals in order to be able to map that variability. The ones in green, are ones that are what I call post hoc aggregate data sets, right? So these are data sets where 20 or so PIs got together and say, well, let's pool all of our resources to build a bigger database, right? The problems with these tools the problem with these things, even though they're great initiatives, they're based off of money that's already spent, right? So you don't have to spend any more money, and these data collections are huge. But the problem is since they weren't coordinated from the beginning, there's a ton of variability there, right? So we're talking about variability in how the data is acquired, the scanners used, the sequence parameters used, how long the scans are, the resting state instructions, whether they have their eyes open, their eyes closed. But also, if we're looking at those other variables we'd like to correlate, IQ tests, the actual IQ tests they were given and how they were administered would be different, right? The exact diagnostic criterion that was applied to say that they're ADHD, right? Your interpretation of those different things. So there's all of this variability that's in there. So there's all of this, sort of quote unquote dirtiness, right? So all of these other things that aren't boxed, these are things that were designed from the top down, right? So these are things where all the participants got the exact same scan. They even may have been at the exact same time of day. They had the exact same thing for breakfast. I mean, I don't know if it goes to that detail, but it goes to a lot of detail, right? If it is multi-site, like the Brain Genomic Superstruct project, they actually have, human phantom aspects of it to actually measure the variability of all of those things, right? So the nice thing about things like, and probably the Human Connectome project is the best example of it, is that when you have that level of homogeneity, it helps you out a lot, right? So if you can simultaneously design your acquisition and your analyses try to be optimized, and you can do extremely well, right? The problem is your analysis probably isn't going to work very well, or may not work very well for anything else, right? Or as well, right? So the Human Connectome Project, they have their own processing pipelines that they release, and they're very good if you collect data the way that they collect it. And you collect the same data that they collect, right? So they have a T1 and a T2, and but they always have field maps, they always have these. If you're doing something with any of these green boxes, though, you don't have that luxury. So what you need to do, what you need are tools that are robust to different pre-processing, to different, say, T1 contrast levels, to different types of fMRI data acquisition, all of those things, right? And so that's one of the major issues with C-PAC. One of the challenges we have with developing C-PAC is that we operate in the regime of the green boxes. So the tools that we build, we emphasize those things that will work with the most data that we've seen and that's available publicly, right? So sometimes people say, oh, well, why don't you have, for example, we don't have field map correction yet, right? We've under-prioritized field map correction because other than the Human Connectome Project and NKI, none of these other data sets have field map correction, right? So we're trying to make a tool that will work with as much data that is, as we believe, is currently being collected. So some of the things are a little bit more exotic in terms of acquisition. We don't handle right now simultaneous segmentation of T1 and T2 like you might get with Human Connectome Project. But it's because we're trying to be robust with as many different data sets and prioritizing that, and then hopefully eventually we will get to some of the other things. But I want to we ought to keep in mind that the ultimate goal that we're trying to do is we're trying to build a tool that will enable processing of these really huge data sets, but also we want it to be robust across those data sets. So our goal is to use algorithms that can be as automatic as possible, right? So that, but of course you always have to check your data. So that we'll talk about that too. So C-PAC is a pre-processing pipeline, and one of the major issues we have to deal with is nobody really knows what the best way to pre-process fMRI data is, right? The irony is with ETI, people believe they know a little bit more about it, even though that's just crazy off the hook in terms of the noise sources, and people don't even understand it. But as we've learned more and more and more about resting state functional connectivity, people have seen like different aspects. There's different physiological noise that people thought was driving functional connectivity, and we have to remove. And then people start using, started using correction algorithms to correct for those things. They say, oh, well, these correction algorithms, they induce chit differences, And then you have other physiological. And you have other physiological things that might affect you, like head motion, that might drive effects, right? So, for example, with ADHD and other hyperkinetic populations, the differences that you see may be exclusively related to head motion, right, and not related to actual sort of neurological relevant or neurobiologically relevant differences in brain function, right? So we have to be very careful about how we process our data to remove all the confounding variation without introducing any sort of bias in the result, right? And so this is another challenge added on to the fact, again, that this is post hoc aggregated data sets. You're going to have a lot more of this variability, right? So not only do you have to remove the variability due to head motion and physiological noise, you also have to try to figure out how to remove variability for different scanner types, different TRs, different TEs, different flip angles, all kinds of things of that nature, right? And so these are the kinds of problems that C-PAC is trying to solve. So on top of the different pre-processing strategies now, there's a ton of ways you can analyze, do individual level analysis for resting state data. So I believe that at last count, C-PAC can generate on the order of 24 different types of derivatives from a single resting state fMRI scan. Right, so we'll bring in, pump in a data set that's maybe 150 megabytes, and when we're done, it's about a gigabyte, right? Because there's so many different things we can calculate from it. And so here's just a couple of the examples that we have, like degree centrality, ReHo, voxel mirror homotopic connectivity, all of these different things. But now, so if you can imagine actually trying to do this yourself, and I showed this before, if you can actually imagine trying to do one of these analyses using just your basic tools, you sitting in front of your computer to do this. If you have 1,000 data sets, it takes about 2 hours to pre-process your data. That's a whole year of work, right? That's 2,000 hours. One human year, one man or woman year in it. that's a lot of time. That's a lot of money in salary. That's a lot of dedicated effort to one project when we're all paid to work on 18 projects simultaneously, not just one, right? So that's a lot of problems. So what you can do though is well, you could script that processing, right? So you could write a bash script to do that processing while you're at home sleeping or going wherever. And so that takes your 2,000 CPU hours and reduces it to 84 days, right? So that's assuming you don't have to interact with the script and it's just running, so that's nice, but still, come on, 84 days, right? That'd take you forever to get your science paper out. But then what we can do on top of that is we can start using high performance computing. We can use multi-threaded processors. We can use clusters, Beowulf clusters, or other clusters in order to run these things in parallel, right? And once we do that, we can start getting things down to minutes, right? Maybe an hour to do 1,000 data sets, maybe 4 hours, a much smaller number. And that's kind of where we want to be, right? The problem is as you go down this list from top to bottom, the complexity of actually implementing what you want to do goes up, right? So you start needing, once you move from, it takes a whole lot of knowledge, technical knowledge about resonance data, fMRI data, and the tools that you're using just to be able to do the 2,000 person hours, right? So you have to be a trained, fMRI person to do it, right? Then once you get to the scripting, now you need to know how to do computer programming, right? And then if you want to do the multi-processing and those things, now not only do you need to know computer programming, but you have to know it deeply enough in order to be able to deal with some complex issues when it comes to parallel processing. Right? So our goal is to do that, to create a tool that provides all of that to you, right? So it does big data. It allows you to use massive high-performance computing. It works with robust systems, and you don't have to have a PhD in computer science in order to be able to pull it off. That's our ultimate goal. Actually, hopefully, you don't even have to have a bachelor's in computer science to pull it off. Our goal is to make it available to as many different people as we can, right? And that's our solution to C-PAC. So just to give you a little bit, and of course, one of the things I put down here is also based on the number of different derivatives you have and different preprocessing strategies that you can do, that actually blows this up a lot more, right? Because you might want to say, well, I don't know how global signal regression is going to affect my data, so let me do it with and without it, right? Well, so now everything's 2. So now 2,000 goes to 4,000, right? So anyway, so that's another part. So our solution to this is C-PAC. So C-PAC was originally created based on by my boss, Michael Milham, based off of his experience with the 1,000 functional connectors project. So if y'all don't know, and I imagine that you probably all do know, but the 1,000 functional connector was probably the first massive large-scale resonance data fMRI data set that was publicly released. And so it's actually more than 1,000 individuals. It's about 1,400. And Mike Milham and Bharat Biswal, they convened all of their collaborators and friends out there and asked each one of them to donate 20 to 30 data sets or more, whatever they were willing to donate, so they could build this massive data set. Well, as a part of that, they did their own analysis of it, and that was in the PNAS paper. And instead of just releasing the software, the data, they decided to release all of their scripts that they used for that paper because they felt like they didn't really think that necessarily people would want it or use it, but they felt like it would help people replicate it, understand what happened, all of those things, right? And I believe at this point, those scripts have been used and cited more than the actual data, right? And that really exposed us to this idea that people really do want this, right? People really do want these tools that they can download from a trusted source. And we're not saying that we are the experts, but we're saying, we're not saying that we know it better than everybody else, right? But we do have enough expertise that we're doing it at least good enough that we are really confident in it. And we're constantly reevaluating things to find out if they're better, right? So based on that, Mike had this idea. It's like, okay, well, we should make a software product that we make that's more sophisticated than these bash scripts we had. And then I talked him into using Python because he wanted to use MATLAB. What good would that do anybody, you know? But so to give you an idea, so C-PAC is a pipeline, right? So, there's tools like Lani, there's tools like NiPipe that allow you, PSOM's another one, that allow you to construct pipelines, right? But again, in order to be able to do that, you have to have the expertise in writing that code and doing those things. So rather than C-PAC giving you the flexibility to implement anything you like, we implement 1 or 2 things and try to implement them extremely well for you, right? And so it is a pipeline. It kind of looks like this. When I talk about pipeline, this is what I mean. And I know you can't read it, but that doesn't matter. You don't need to know what these are. But just the fact that there's a series of steps that are sequential, right? Some of them can go in parallel. But it's the data is just, step 1, do this, step 2, do that. And that's what a pipeline is, right? So these pipelines can actually become more complicated because at one point we may decide that we want to do it 2 different ways, right? What CPAC will allow you to do, and I'll show you an example in a minute, is to do it both ways, but to only do those things that need to be done twice, right? So you don't have to go back. So if you're say you want to do this one, this process 2 different ways, you don't have to go back here and start and do that twice, right? That's already done. So CPAC is there to hopefully intelligently identify areas where it can achieve speed up by reusing things that are already calculated, for example, right? We try to be the most cutting edge in terms of functional connectivity. So when new methods come out, new analysis algorithms, and new processing algorithms, we evaluate it. And you can look at our publication record and see those evaluations occurring. And then we try to implement those things to determine what's the best. And as a consequence, those are what our users now have available, those different things that we've evaluated. At the end of it, we have a recommendation for what's best, but you also have other alternatives that we've implemented for it. Currently, CPAC is probably, the most extensive set of resting state fMRI analyses and preprocessings that are currently out there and pulled together. So we don't reinvent the wheel. Mostly CPAC is a ligament that connects other programs together. We do write some of these algorithms ourselves because they're not available anywhere else. But we rely on other tools that y'all would commonly use and are probably already available, knowledgeable about, like ANTS, AFI, and FSL. So as I mentioned before, we have this ability to do things multiple different ways so that y'all can evaluate different things, and we can do that with minimizing the computational penalty of it. Once we create this workflow graph for a pipeline, we can go through that graph and we can identify areas where parallelization ability exists, right? So, and then we map that to your architecture. So you tell us what you have, and say you have a SGE cluster. Then we can take, and each one of those nodes has 8 processors on it or whatever. Then we can take what you've given us and we can come up with a strategy to parallelize it automatically to make it more efficient, right? And really all you've had to do is tell us answer some very basic questions about what your architecture looks like. And some of those harder questions, you just ask your system to fill it out for you, So it can be very, very simple that way. Also, we allow this ability to do warm restarts. So the idea is so you may go through a pipeline and it terminates for some reason. Maybe you run out of memory or whatever. Or maybe you go through and you decide, oh, well, I wish that this parameter had been different. And so you stop it. You can do a warm restart, which means CPAC searches and identifies all the things that don't have to change based on whatever you've done, and only recomputes those things that do have to change. So the idea is it can make it so you don't have to start from scratch every time you redo it. We're making a lot of leaps forward in terms of making it highly efficient, though. So maybe it doesn't necessarily hurt you to recompute some things that override. But also a big thing about it is it's open science, it's open source. Everything that we have is openly developed on GitHub from our development pages. So y'all can see it as it's happening. You can look at our implementations to learn from them or to critically evaluate what we've done. We're very happy to hear any criticisms that people may have. And it's free, which is nice. Okay. So this is our webpage at the end, and I'll show you the webpage. But I wanted to talk a little bit about forking. So here's a forking example. So the idea is that, so here's the basic preprocessing. So this is cut down a lot. We've reduced a lot of the nodes here. And this is, again, I'm sorry, a little bit hard to read. But so we have our anatomical data processing that needs to happen. So when you process resting state data, you also need to process anatomical data. And so that can happen, and there's some basic processing that needs to happen on our fMRI data. And those things can happen in parallel for the most part, right? There's only a couple of places where you need information from the anatomical to do the EPI. So CPAC would identify these things as being parallel and execute them in parallel, right? So we get down to this one point where we go through nuisance correction, and we'll describe that a little bit more later. But we go through nuisance correction, and we have bandpass filtering. So in one pipeline, we have bandpass filtering, and then that makes it blue. And then the other one, we don't have bandpass filtering. So when we go to the page that it says filtering, we can select on, off, or on and off, right? And if it's on and off, we fork our pipeline. So now we have so everything past this line was done twice, right? So it's more efficient in the terms of you don't have to do this again, but also it's efficient in the terms of these things can happen in parallel, and we identify that parallelism and we exploit it in order to make it faster. But you'll hear me say forking a lot, and that's what I mean. We also have another concept that's called an iterable, and that's when you have multiple parameters, right? So say you want to do, for example, with bandpass filtering, say you want to use different cutoffs, right? You can give a list of cutoffs, and it will do it once for each one of those cutoffs, right? So here where you have this fork that goes into 2, it'll actually go into as many different cutoffs as you have. So say you had put in 10 different thresholds, then you would have 10 there. So using these two techniques, you can blow this up pretty big, right? So imagine if I did put in, I had bandpass filtering on and off, and then I had 10 things, or 10 thresholds for the bandpass filtering, then I would have 11 different pipelines here, right? 1 where it's off and 10 where it's on. But then if you imagine there was another decision after filtering that we had on and off, now you would have 20, right? So these things can blow up pretty quickly. So I always encourage people to keep that in mind when they're doing it, because we would like to just be able to really explore the space, try a couple of different things, see the effect of it. But even with something that can run efficiently, you'll end up bogging yourself down. Another thing, and this is something y'all have heard about, is that we're now supporting computing in the Amazon cloud, and we'll go over that. But I've gone over that quite a bit. It's just a way that y'all can have access to high performance computing that isn't currently, if you don't currently have those resources. So those are. So that was kind of a brief overview of why we created C-PAC and kind of what C-PAC is, maybe in a nutshell. Do y'all have any questions? No? Do y'all want to take a break, coffee break or anything? Okay.
Maybe yes.
Yes?
Yes, maybe. Just
okay.
It's just to give you because Raul, the director, came and took 2 of our main users.
Okay.